In this task, I'll be demonstrating an unsupervised machine learning model with k-means clustering, the purpose of which is to identify the clusters in the given dataset. The dataset under consideration here is iris dataset, which I imported from sklearn datasets. The other libraries I've used are pandas, numpy, and matplotlib for performing necessary calculations and visualization. After importing my dataset, here is how it looks like. There are actually four features, namely sepal length, sepal width, petal length, and petal width. I've concatenated all of this to form a data frame. Then I assign features to X and target to Y. For finding the number of clusters in the dataset, I've used the elbow method. So I consider dataset in the range of 2 to 10. Then I plotted the average distortion score for each of this value. And the plot shows the shape of an elbow, which is why it's called an elbow method. The point where there is a maximum inflection in the graph will give the best value of k. That's why I've chosen k to be 3. Then I assign k as 3 in my k-means algorithm that's here. Then I plot the graph. So from the plot, it's evident that there exist 3 clusters, cluster 1, 2, and 3, which are distinguished by 3 colors, green, blue, and red. And the yellow dots indicate the center of each cluster. What I notice here is that there is an overlap in between cluster 3 and 1. So I thought of performing a principal component analysis. What a principal component analysis does is it reduces the dimensionality of our dataset. So let's take a look at the dataset. So here we have 4 features. So when I specify n components equals 2, it reduces the dimensionality. This is done for simplicity. So this is our new features, xp. Assign here, xp here. And this is the, xp. So this is the plot that I obtained. So there is a more distinct separation between the red and blue colors here. And what can be inferred from this study is that there are 3 clusters which exist in the iris dataset, which are actually the 3 species of iris flower, namely Iris verticolor, Iris virginica, and Iris setosa.